Thank you, Tian. It's great to be on with you.
Oh, great, Paul. Thank you. And really excited to hear about the company and your updates. We'll go ahead and get started.
All
Welcome,
right.
everybody. I'll make a quick introduction and then turn it over to Paul. Welcome, everybody, and thank you for joining us for this awesome presentation by Angiex. Angiex is developing a new class of antibody-drug conjugates designed to deliver therapeutic payloads directly into the nucleus of tumor cells and tumor-associated vasculature. The company has a lead asset called AGX-101. It's targeting a protein that's highly expressed in tumor blood vessels and tumor cells with limited expression in normal adult tissues. Company is currently in phase 1 trials with this AGX-101 with patients with advanced solid tumors. It's demonstrated exceptional safety profile so far, alongside early signs of clinical activity, including stable disease and tumor shrinkage at higher dose levels. What differentiates Angiex? A couple things. First of all, it is a first-in-class nuclear delivered ADC platform. It also has validated tumor target, TM4SF1, which enables dual targeting of both the tumor vasculature as well as the tumor cells themselves. There's 3 complementary mechanisms of action: vascular deprivation, immune activation, and direct tumor cell killing. Another differentiator is already they've got an outstanding safety profile with no observed adverse effects in primate studies at the highest dose tested. And lastly, there's early clinical validation. So on target pharmacodynamics and emerging tumor response in phase 1, 5 great differentiators for the company. Without further ado, I'd like to introduce you to the founder and CEO, Paul Jaminet. Paul, welcome, and thank you for sharing today.
Yeah, thank you so much. It's great to be on. That was a very good introduction. So Angiex, it's a very exciting company built on a very exciting biology. It's the fruit of 23 years of biology research and 13 years of drug development. Our scientific founders were leaders in the field of angiogenesis. That's the formation of new blood vessels. And the holy grail of that field is finding a universal therapy for all growing tumors that works by eliminating the growing vessels of the tumors without harming the quiescent vessels in the rest of the body. So our founders devoted their careers to that, and they discovered a way to do it through the biology of a protein TM4SF1, which is still very lightly studied today, and our founders have done almost all of the key work on it. But it turns out to be great for antibody-drug conjugate therapy, so we made a drug against it. On the right, you can see a picture of our antibody-drug conjugate. So in blue is the antibody that provides homing to the tumor, and in yellow is the conjugated payload. That's what gives us efficacy against the cells in the tumor. And as Tian mentioned, we can kill 2 parts of the tumor, the blood vessels and the tumor cells, and that gives us 3 mechanisms of action. We're now in phase 1 clinical trials. We've successfully prevented tumor growth in all tumors, primary or metastatic, over 40 tumors from 14 different cancers across 3 different dose levels. So I'll get into efficacy and safety a little bit more later. But the investment opportunity, you can enter now at a valuation that's fairly typical of oncology companies that have not yet generated positive clinical data. I think with us, you can see that the very exciting data is coming very soon. But there's a huge value inflection that's imminent. And after approval, comparable companies are valued about 1,000-fold higher. So big opportunity. So this gets to some of the biology, why this target is so exciting. So first of all, it's essential for blood vessel formation and for blood vessel growth. So vessels in the tumor cannot grow without it. And that means that all tumors are going to be sensitive to our drug. Uh, Likewise, we have a more or less universal method of turning tumors immunologically warm, which can generate great responses to immuno-oncology approaches. And then tumor cells can't invade or metastasize without it. And so the most malignant tumor cells are generally sensitive to our drug. And in adults, because TM4SF1 its function is to enable growth, tissue growth. It's really not active in normal tissue, but in tumors, it's highly active. So you can see very abundant staining of the tumor cells and of the endothelial cells of tumor blood vessels. And that means that on clinical radioimaging, the tumor really stands out compared to other organs, which is not the case for other oncology targets. And so this outstanding tumor homing really gives us the opportunity to be effective against the tumor and very safe in the rest of the body. And this slide illustrates why this target is so good for antibody-drug conjugate therapy. So in green, you see our antibodies labeled with this green dye. And this is what the cell looks like just after it's been exposed to our antibodies. Each of these little green dots on the cell surface you can think of as a freight train that starts its life unloaded with no cargo on it on the cell surface. And its function is to load up with growth factor activated proteins that act as transcription factors and transport them to the nucleus so that they can regulate gene expression. And that's what it does. So what you see here, it's been loaded up with our antibody, but it's also loading up with growth factor activated proteins. 4 hours later, this is what the cell looks like. So all of these freight trains have loaded up with cargo and they've internalized. And first they head to one side of the nucleus to a place called the microtubule organizing center. That's like the grand central station of the cell. And then they go through nuclear pores into the nucleus. And in the nucleus, our antibodies are degraded, and that releases our payload. And the nucleus is a fantastic place to deliver a drug. So, great opportunity. We have this antibody that homes to tumors and mostly ignores the rest of the body. And then it internalizes to the nucleus and drops off this conjugated payload in the nucleus of the tumor cells and the tumor vascular endothelial cells. And we've been the only people working on this for 23 years, and there was essentially no prior art. We're the first ADC to enter the clinic. And so as a result, we've been able to build a very strong patent portfolio. We have 12 patent families based on preclinical data and the drug. And we're about to file a couple more based on clinical data. So the next few slides show how we get efficacy. So this slide illustrates the attack on the tumor blood vessels. So the drug eliminates the blood, all the small vessels within the tumor, but it also eliminates the large vessels that feed and drain the tumor. So that there's feeding artery, draining vein. Uh, and, and that's like shutting off the master water valve to a house. No one inside the house can drink. So when we eliminate these vessels that feed the tumor, none of the cells inside the tumor can get nutrients or oxygen, and so they die. And you get a necrotic center, which the immune system then tries to heal or clean up. But the result is that we can kill a lot of tumor cells. And so this is a universal mechanism because no tumor can grow without expanding its blood supply, and the attempt to expand its blood supply makes the tumor sensitive to our drug. What this slide illustrates is that when we injure the tumor vessels, we convert the immune profile to an immunologically warm profile with cytotoxic T cells, antigen-presenting cells, natural killer cells, And that leads to a very good response to immune checkpoint inhibitors. So what we found is that we can combine with either CTLA-4 or PD-1 antibodies, and we can convert the tumors from immunologically cold to immunologically warm and make them respond to the checkpoint inhibitors at a high rate. So typically 70 to 80% tumor-free response rates, even in models that are not responsive to immune checkpoint inhibitors normally. And the great thing is this works at very low doses, around 2 milligram per kilogram, which are extremely safe for our drug. So, great potential. These checkpoint inhibitors sell $50 billion per year. They're going off patent, and they only work in 12% of patients. So we can potentially make them work in 70% of patients and own all the patents on the combinations. So a great opportunity there. And then we can kill off the invasive and metastatic tumor cells because they express TM4SF1 at high levels. And so we're effective against by tumor cell killing in a very broad range of models. And this slide illustrates that because tumor cells need TM4SF1 to invade and metastasize, in late-stage cancer patients, most of their tumor cells are high expressors of TM4SF1, and that's true across all solid cancers. So here in blue, you see the expression level of TM4SF1, and it's plotted against the expression of the targets of other approved solid tumor antibody-drug conjugates. The most successful other target is HER2. That's shown here in orange. And HER2 is higher than TM4SF1 in breast cancer, but it's lower in every other cancer. And in about half of all solid cancers, TM4SF1 is higher than HER2 is in breast cancer. So, great potential for this tumor cell killing mechanism to really have a huge impact on the tumor cell populations in these patients. So all of these things generate a huge addressable market. So the anti-angiogenic approach is, as I mentioned, a universal approach. We can attack every growing tumor through this attack on the vasculature. And prior anti-angiogenic drugs have sold very well, even though they weren't very effective. So, for example, Avastin had revenues of about $100 billion, even though it only extended survival by about 2%. But even though it had minimal efficacy, it combined well with other drugs. Its toxicities were non-overlapping with other oncology drugs, and so it was very easy to prescribe. We also have that benefit. Our tox is in different places than other oncology drugs, so we'll sell very, very well. And I mentioned, huge market for immune activation and combination with checkpoint inhibitors, and then this tumor cell killing can deliver a clinical benefit in all solid cancers. And as Tien mentioned in his introduction, we were very safe in animals. So this plot shows you the blood concentration of the drug in black that's needed to shrink tumors. And in blue, that's a no observed adverse effect level in healthy monkeys. And there's almost a factor of 100 between these. And this is really the best predictor of clinical success and of a fast course through the clinic and capturing a large commercial market. The reason for that is that there's going to be a lot of heterogeneity in patients. So there's heterogeneity in safety. So imagine this curve blurred out, and there's heterogeneity in efficacy. So imagine this curve blurred out. But if they're widely separated, that means you can find a dose in the middle where you're both effective and safe in just about everyone. And so that's our aim, is to figure out how to help every cancer patient. There is prior clinical experience, very promising. They had 100% response rate in patients dosed 3 or more times, and they had negligible off-tumor toxicity, apart from off-target toxicity from the radioactivity. So this indicates that there's every reason to expect our drug to do very well. And so this is where we're at in the clinic. So we've completed the first pass through our dose escalation. We dosed 3 patients at the highest dose level. This is a higher dose level than any tubulin inhibitor ADC has previously reached. And at this dose level, 2 of the patients had only mild adverse events. One patient had a severe liver injury. So we haven't determined yet if this level is tolerated or not tolerated. But this lower level is well tolerated. And we've started to explore different dosing intervals at that dose level as well. And we've started combination arms with immune checkpoint inhibitors. So we've dosed 3 patients in combination with Keytruda. And this is just a look at what we've seen in the clinic. And so what we see is a universal wounding of tumors. So we wound all the tumor lesions. And I've got 2 slides here just to show you the effect of the lesions, of the tumor wounding on CT scans. So we kill a lot of tumor cells as part of this. But you can see this was the original lesion. This is the patient with metastatic ovarian cancer, and this is metastatic tumor in the liver. And you can see there's a slight change in structure, but after 2 doses, uh, what you see is it looks a lot more like background liver. It has a mottled, the lesion now has a mottled appearance. It's gone from being these ovarian cancer cells, they're very liquid. They kind of resemble, you can see they're less dense than the surrounding liver. And that's a characteristic of ovarian cancer cells. Here you can see the density is increasing. So we think this lesion is now largely a mix of scar tissue and cavities, so where there was a necrosis and the cells were cleared away, you can get a cavity. But if the matrix survived, it can be collagenized and turned into a scar. And then here's another lesion in that same patient. And in this case, the tumor became the tumor lesion. This is before dosing, so a tumor. And then it got smaller, but it also got blacker, meaning that it was less dense. And this uh, level of blackness is similar to what you see inside the stomach or inside the colon. And so we may have made a cavity where the tumor was. So uh, the most definite thing we've learned from this experience was that CT scans aren't that good for helping us manage the patient because they seem to leave a lesion that's approximately the same size as the original lesion, but looks like it may consist of a combination of scar tissue and cavities. But it looks different than the surrounding normal tissue, and so the radiologists still see it as a lesion. So we don't really know how many tumor cells have we killed? Have we killed all of them? Should we stop dosing? Should we continue? And so what we've done is we've introduced PET imaging to the study, and we're redoing key dose levels. So if I go back here, we're redoing starting at 3. We'll do these highest 3 dose levels over again with PET imaging in order to understand how best to treat the patients. But we're homing in on the best way to treat. We've identified our lead indications for approval. The top 2 indications are angiosarcoma and pancreatic cancer. And I'll just briefly speak about angiosarcoma. It's a rare cancer, but that has some benefits for development, including very fast tracks to approval. In angiosarcoma, there's an especially easy regulatory path because there's no approved therapy for angiosarcoma currently. And the standard of care doesn't work. So the second line standard of care, the response rate's 5 to 9%, median progression-free survival 3 months. and median overall survival 13 months. So this is the bar to beat for accelerated approval. This is the bar to beat for full approval. And if we have, as we expect, a 100% response rate and an indefinite progression-free survival, then you can meet the bar for approval in a 25 patient trial in 3 months. So the bar for accelerated approval and for full approval in 6 months. So great potential to really bring uh, the drug to approval quickly and help a lot of patients. And so this is just where we're at as far as site activation. And we have a number of top cancer centers coming on, including MD Anderson in Houston, Dana-Farber in Boston, Washington University of St. Louis, the University of Southern California in Los Angeles. And these include top cancer centers for angiosarcoma. So collectively, they treat about 40% of all the advanced angiosarcoma cases in the US. And so this is a sufficient site network to bring our drug to approval. The path to approval looks like this. So we're currently in this blue period. We're figuring out how to dose, trying different dosing regimens and demonstrating that the drug works. But once we figured out how to dose, then we can go on the clinical side. We can go to these phase two expansions, and we can first clarify exactly what the dose of the label of the approved drug should look like, and initiate a pivotal trial to generate the data for approval. And in parallel, we can complete building our commercial supply chain and meeting all the regulatory requirements for approval on the manufacturing and quality side. Once we have all the data together, we can complete the registrational trial, submit all the applications, and get the drug approved. So this is the timeline. In the first half of 2026, we're completing that blue box to prove that the drug works and figure out the recommended phase two dosing. We can do these start expansions in the second half of 2026, and then a pivotal trial will be in 2027. And then we can submit our application for approval in 2028 and hopefully get approval before the end of 2028. And this plot shows how valuations go up as you progress through the clinic with a successful drug. So the post money on our last round, last financing was 50 million. And that's in line with companies that don't have successful drugs. But once you prove that you have a successful drug, which we hope to do in 6 months, then the average oncology company is valued at 400 million, and ADC companies have often been valued north of $1 billion. And then once you're approved, ADC companies have been consistently valued north of $10 billion. So our financing strategy will be tied to valuation. So we're currently raising a top-off financing or bridge to additional data. We'll accept up to $10 million. And we expect with that money to reach this 10x or so value inflection to $400 million or so. And from there, we can raise the money to get the drug to approval and the company to profitability. And that will be done over a series of financings, so maybe three $50 million financings, one of which we expect to be an IPO in 2027. And then we expect to get to revenue in 2028. So just a little on the team. I myself am started my career as a scientist, an astrophysicist at MIT, Berkeley, and Harvard. Then I became an entrepreneur during the internet boom. I founded four companies, written a best-selling book, I'm aided on the executive side by Ian Dukes, our executive chair. He led business development at Amgen, led business development at Merck, and as a venture partner at OrbiMed, served as CEO or chairman of a number of OrbiMed companies. Our chief medical officer led oncology research at one of the Harvard hospitals. He's done over 150 clinical trials and been a chief medical officer prior. Our CMC team, Brian Clark and Morris Rosenberg, led manufacturing and quality at the top ADC companies in the country, the top East Coast company, Immunogen, the top West Coast company, Seattle Genetics, and between them, 7 approved ADCs, $70 billion of corporate acquisitions. And our Chief Financial Officer, Jeff Satcher, is on the call. Jeff, do you want to give a quick introduction?
Sure. So five-time CFO, some of them are listed here. And then prior to that, spent 25 years in investment banking, Goldman Sachs, Brean Capital, B. Riley, National Securities. So worked with other oncology companies as well as a CFO. And that's my background. Look forward to dialoguing with folks.
Yep. And our scientific founders made the top two discoveries in the field of angiogenesis. One of them, VEGF, was the basis for Avastin, $100 billion of drug sales. And then my wife discovered our biology, so that's how I learned about it early on. And TM4SF1 will be a much bigger value creator than VEGF was. So exciting opportunity. And, so as I mentioned, we have a current top-off financing of, we're raising $10 million. We'll accept small checks, $50,000. If that's too much, then reach out to me and talk to me. Everyone should try to invest as much as they can, both because it's a fantastic investment opportunity and because you can have a big impact. The main goal of this current fundraise is to fund this redoing of the upper dose levels of the dose escalation with PET imaging to get a tumor cell count and figure out the best way to treat and help the patients. And just a note, our stock is qualified small business stock, and so capital gains can be tax-free. So that's a great advantage. And in conclusion, I just like to say there's a lot of people who need our drug. One person who would have been helped by it was my mother. She was diagnosed with cancer at age 22 when she was pregnant with me. She died at age 33 when I was 10. And I keep that picture right next to my computer monitor, so that I see it all the time. And it reminds me that there's a lot of people depending on me. I have to work hard. But the thing that limits our rate of progress, our speed to approval, is capital in. So the quicker we raise money, the more aggressively we can spend, and the quicker we'll get to approval, and the quicker we'll be able to save lives. So I hope you'll come in and join us, and it'll be an exciting journey, I promise.
Great. Thank you so much, Paul. We're going to do a couple things. We'll launch our investor poll. This is confidential for those of you attending today. If you are very interested in the company, you want to set up a follow-on meeting. If you're interested and want more information, if you are not interested, but you have some investors who might be interested, or if you're not interested, if you don't mind responding, we will keep these responses confidential. We won't share them with you guys. We'll share them with Paul, obviously, and Jeff. But we can kick off our Q&A session here. We have some questions already. If any of you have any questions for Paul or Jeff, please either type them directly into the group chat, or you can private message Emily or myself if you don't want us to attribute the question to you, or raise your hand and you can ask. So with that, the first question, Paul, we have is, can Can you walk us through the clinical data for AGX-101 to date, as well as the evidence supporting efficacy? I know you touched on it briefly in the deck, but if you go a little deeper.
Yeah, yeah. Um, well, I did cover the gist of what we've learned on the efficacy side. So what we've seen is a universal effect. Essentially no lesion has grown beyond its baseline size. So I've actually shown you here, these are the two most extreme. This is the tumor that shrank the most, and the one above it, this is the lesion that grew the most. So every other lesion, has been, even closer to its original size. And you can see, you know, when they grow, they get less distinct, and when they shrink, they get more distinct. But what we think is happening is that the lesions are being converted to some combination of scars and cavities, uh, scar scar tissue and liquid-filled cavities. And so we weren't aware that that would be the outcome because it didn't happen in mouse models. Uh, and so we were relying on CT scans, which is the standard to assess whether tumors shrink. And it looks like we're killing the tumor cells, but there's still a lesion, and so the doctors don't know what to do. And so we're adding PET imaging and redoing some dose levels. And then on the safety side, what we found, this dose level 4.5 and all the dose levels below it have cleared safely. So we've dosed 11 patients at 4.5. We've dosed, I think, 7 patients at 3. And we've dosed 3 patients at this 6.7. This one has not yet demonstrated safety. It's not cleared, but it's also not been untolerated. So what happened is we had 1 of 3 patients have a severe adverse event. That was a case of liver injury. And the margin to prove safety is you have to demonstrate with statistical significance that the incidence of those severe adverse events is below 30%. And 1 out of 3 is close enough to 30% that it's considered that what you need to do is add more patients. But we wanted to go back, since we're introducing the PET imaging, we want to go back to a lower dose and see what happened there, because at 3 milligram per kilogram, we also didn't see tumor growth. So we think we were killing a lot of tumor cells from at least 3 milligram per kilogram upward. And so the ideal dose could be anywhere in this range. And so we're going to explore it again with the PET imaging. And we've more or less settled on a Q6-week schedule as the optimal interval, but we'll still do some further evaluation to prove it. And we've more or less figured out how to scale the dose by body weight, which is an important thing to determine. So I won't give details because that's the subject of an imminent patent application. But a number of drugs failed because they used the wrong scaling. And so in obese patients, they would have a higher incidence of adverse events. And we think we've figured out the right way to scale by body weight. So we're homing in on all those elements of the drug label that you need to get to approval. So I would say we've accomplished all the things that other companies generally accomplish in their phase 1 dose escalation. So we found a safe range of doses. Uh, and so most other drugs, they don't expect to show efficacy in their phase 1. So they do the phase 1 to identify a safe dose, and then they home in on a very specific patient population to prove efficacy. But in our case, we appear to have universal efficacy. So the drug works apparently the same on every tumor, regardless of whether it's primary or metastatic and what cancer it originated from. And we just didn't have the right tool for assessing efficacy. Uh, we need the metabolic measurement given by FDG PET rather than look at the tumor matrix given by CT scans.
Got it.
Um,
Thank you so much, Paul. Oh, sorry. Go ahead and
yeah, go ahead. Go ahead.
yeah, speaking of IP, well, before I ask the next question, we're gonna leave the poll up another 30 seconds. So for those of you, I think a couple of you have not filled out the poll yet, you can either X out by hitting the red button or you can answer, but we'll keep it up for another 30 seconds. Again, it's for very interested, not interested, but have investors who might be interested or not interested at all. Speaking of IP, we have a question here. Does Angiex fully own the intellectual property for AGX-101? What is the remaining life of the key patents, and how does that translate into market exclusivity?
Yeah, yeah. So we do own all the IP. So we developed the drug entirely ourselves. We have freedom to operate. There's no royalties owed and no licenses. It's been a huge advantage being the only people working in the field of ADC, TM4SF1 directed ADCs. As I mentioned, we have 12 patent families. We have some patents that protect broadly against any TM4SF1 directed ADC. So we were able to increase therapeutic index tenfold over the only prior preclinical ADC that had been made. And we were able to patent the means to do that. And so we think we're protected against competition, against all TM4SF1 competitors through about 2039. So those patents were filed in 2019. Our picture patent, which is the composition of matter patent, was filed in 2021, so that protects us through about 2041. And then, as I mentioned, we're filing new clinical patents now, which will give us further protection against, both all competitors and especially against biosimilars. And so those should bring us to 20 years from now, which is 2046. And then a lot of our indications, including our first two indications, angiosarcoma and pancreatic cancer, are orphan eligible. So we can get orphan designation in the US, which adds 7 years of exclusivity. And there's some similar exclusivity benefits to addressing rare cancers in other jurisdictions as well. Very strong patent moat and a very long, we think we can get to approval in 2028 or 2029. And so with a strong patent portfolio extending through 2046, that gives us an exceptionally long period of market exclusivity for pharmaceutical drugs.
Great. Thank you, Paul. Next question. Have pharmaceutical companies approached Angiex regarding AGX-101, and how would you characterize those discussions?
Yes, they have. And so I would say, they're waiting for clinical data. They're waiting for proof that we're killing off the tumor cells and shrinking the tumors. So they're not necessarily super innovative when it's a novel target that hasn't been studied before. They don't want to guess what's going to happen or rely on their scientists to make predictions. They want to see clinical data. But I would say, people are fascinated by the huge potential. They can see that it can be transformative. And so they want to follow us. They want to see our data when we have it. And, uh, so they don't tell us what internal work they might have done to evaluate the target, but they're definitely
Mhm.
want to see our clinical data when we have it.
Got it. Have a question in the chat from Raj. What stage does this drug typically target?
Uh, well, it will work at all stages, so actually the earlier the stage, then the more complete the treatment and the fewer, adverse effects patients would notice. But I would say our quickest path to approval is in these late stage, high unmet need cancers. Patients who have metastases, they have few options. Uh, when people are close to death, it's very easy to generate a survival benefit and meet the requirements for approval. So that's where we'll start. And also in the phase one patient population are those late stage cancer patients. Those are the ones who are enrolling in clinical trials and looking because they've failed all the approved therapies. Uh, so right now our focus is on those late stage patients. But once we get to approval in the late stage high unmet need patients, then we'll, gradually expand our range of indications to and go to earlier lines of therapy and ultimately to the frontline therapy. We hope to be the frontline therapy for all solid cancers.
Got it. And speaking of approvals, a couple questions regarding FDA. What are the major steps required to obtain FDA approval for AGX-101 in its first indication? And where do you see the greatest regulatory risk? And similarly, how have interactions with FDA progressed to date? What are the key topics planned for upcoming FDA engagements?
Yeah. Okay. Great question. So, um, so there's basically two major tracks that you have to satisfy the FDA about. And the one that tends to be critical path for antibody drug conjugates is this CMC, chemistry, manufacturing, and controls, and which is basically manufacturing and quality. So what you have to do is you have to put together your commercial supply chain. You have to have your commercial process and your commercial quality assays all in place. The FDA has to have signed off on all of these. And then you have to produce at least three, and in practice, four repeated batches at commercial scale, and demonstrate that the drug comes out the same way every time. So you need to demonstrate repeatability of production. And so that part, this is really the expensive part. It typically for ADCs costs 65 to 80 million dollars to meet these requirements for the manufacturing process. And it also takes about 18 months. And because it's so expensive, you don't generally start it, and we won't start it until you've gotten this valuation lift, to a valuation north of 400 million. Then you can raise the $80 million to pay for this. And then it's 18 months. And at that point, you have the material which can complete your registrational trial because about half the patients in your registrational trial have to be dosed with this commercial quality material. So that sets the timeline and the biggest expense. And then in parallel, you're doing clinical research to figure out all of the elements of the drug label. So the registrational trial is really a confirmational trial where, you're proving that, what you said was going to be the label for the drug is really accurate. And uh, so that registrational trial, you can start it with the clinical material, but you have to finish with the commercial quality material. And so like I said, the manufacturing is the critical path. This will do in parallel uh, with the manufacturing and quality work, and we expect to start our pivotal trial in early 2027. and we'll have the commercial quality material about summer 2027. And so we can get the majority of the patients on in the last half of 2027. And then we'll have our data package in 2028, submit our application for approval in 2028, and maybe get the approval before the end of 2028 or in 2029. So that's the timeline. As far as engagement with the FDA, it's been excellent. So we're on the ninth version of our protocol. We've amended it 8 times so far based on clinical data. We were granted fast track status for pancreatic cancer uh, last year. And I would say our interactions with the FDA have been very positive. And as we finalize things, we'll be having a more extensive meeting into phase 1 meeting where we'll ask them to approve the recommended phase 2 dose that we use for the clinical studies here. And then we'll meet again with them to make sure that our pivotal trial design is what they want. And also, as we gather data, we'll submit breakthrough therapy and orphan designation applications. So those will be, important value inflecting milestones. So investors put a lot of credence on those designations. The orphan designation has a lot of financial value. And the breakthrough therapy designation, about half the drugs that get breakthrough therapy go on to be approved. And so it's a mark from the FDA that they think you have a very high chance of being approved and having an impact.
Got it. Just a quick question here on the size of the tumors that you can address. What's the minimum and maximum size of the tumors?
There's no minimum and no maximum.
Mhm.
The larger your tumor burden, then the more drug we may need to dose because the tumor will soak up drug, or the more doses you may need to receive, because uh, so in the average late stage cancer patient, about 60% of all the antigen is found in tumors. So I showed you that uh, earlier. It's just there's so much more antigen in tumors, typically 20-fold more per cell in tumors than in normal. Uh, just in the vascular endothelium, and then you have all these tumor cells that have a lot of antigen as well. Typically in the late stage patients, a majority of the antigen is in the tumors, and if you have a very large tumor burden, it can get uh, well above 50%. So, um, so if the tumor burden is really large, then it would be like peeling the onion. We may only kill a fraction of the tumor cells with each dose, and some of the tumor cells just consume all the drug, and so we have to shrink the tumor cell population down over multiple doses. Whereas, uh, in patients with uh, so in in these tumors, we may have largely eliminated the tumor cell population just with 2 doses.
Mhm.
So, this is an ovarian cancer patient with 3 metastatic tumors. Um, so late stage, but they're not huge tumors. These were 2 centimeter tumors.
Mhm.
Um, so, so I think it's uh, the impact of stage, maybe how many doses does the patient
Got it.
need, but it shouldn't prevent us from treating anyone.
Got it. So the mechanism of action works. It's just a matter of dosing, right?
Yeah, it's just a matter of, how long does it take to achieve a complete remission? That's going to
Mhm.
vary from patient to patient.
Got it. Okay. Question on competition. Who are your major competitors? How does this drug stand against other competitive drugs?
Um, yeah, well, I would say, immuno-oncology approaches, they're the best-selling drugs in oncology today. And they complemented us very well, but they're also, a competitive alternative. So it will be very valuable to show that when our drug combines with the immuno-oncology drugs, that they work much better. And we think that will be the case because we believe we turn the tumors immunologically warm consistently. So if we prove that, and we get patents on the combination, and everybody wants to take immuno-oncology in combination with our drug, then we've eliminated a whole class of competitors. And then, another major class is antibody-drug conjugates. And, so antibody-drug conjugates are replacing chemotherapies because they can have broader therapeutic margin. Basically, instead of chemotherapy, it's just one small molecule. It has to go all over your body. So it's going to cause a lot of tox and it'll have narrow therapeutic margin. The antibody drug conjugate, because it's a two-part drug, the antibody can direct it more fully to the tumors, keep it off places in normal that are sites of tox, and so you can broaden the therapeutic margin. So the industry is moving away from chemotherapies to antibody-drug conjugates. But as this shows, the other ADC targets, they're on tumor cells, they're not on other compartments of the tumor. So we have two ways to attack the tumor cells and the tumor blood vessels. They only have the tumor cells. And we're better as a tumor cell target because there's a lot more TM4SF1 on the tumor cells, and it's much more consistently expressed. I think we'll compete very well against other ADCs in all solid cancers. And I think for the same reasons other ADCs are replacing chemotherapies and, proving superiority toward them, we'll establish superiority to chemotherapies as well. So I think, our competitive position stands in a very strong place. I would say the oncology landscape is likely to come down to TM4SF1 directed ADCs plus immuno-oncology approaches, and it will probably be predominantly the immune checkpoint inhibitors like Keytruda, Yervoy, Opdivo will be the successful drugs of the future, and all the biosimilars because those are going off patent.
Got it. I have a question about some financials here. What is the valuation for the $10 million bridge round? What is the investment vehicle? Is it a SAFE or a convertible note, or is it a stock sale? Is the lockup period for stock and any other restrictions? What is the lockup period for stocks and any other restrictions if it's stock that's being issued? And what is the exit strategy plan so far? IPO versus acquisition at what stage? That's a lot.
Yeah. All right. Well, starting from the end, we're planning an IPO in 2027. So that was here, let me find that financing slide. And so basically a progression of financings culminating in a 2027 IPO. Right now, we already have an all common stock cap table. So we've started preparing for an IPO, and as part of that, we converted all of our preferred shareholders to common. And that's a very favorable situation for small investors. So there's no powerful players at the table with blocking rights that they can use, that give them bargaining leverage. So everyone's aligned in their incentives. The fiduciary duties are aligned with the shareholder incentives to maximize the value of the common stock and the value of the company. As far as lockup periods, IPOs generally do have lockup periods, but as a private company, the equity is restricted. We can try to find, if somebody needs liquidity, we can try to find and arrange secondary sales, but you should assume that the stock is illiquid until an IPO, and you should assume there might be like a 6-month lockup period after the IPO. But I would say, because it's qualified small business stock, it's in you get tax advantages from holding on to it anyway. So that may not be that big a deal. Um, and then as far as the financing, so we have an open convertible note. And so you can invest in that immediately. That has a 20% discount, a 10% interest rate, and a $200 million cap. And it has optional conversion rights in various scenarios. So the note holders are protected in every scenario. We will accept equity term sheets from anyone who is investing at a size of $1 million or higher. So we can negotiate a price. So we'd expect it to be higher than the post money on the last round, which was 50 million, but we can discuss that. And in addition, there's an SPV with no fees, no carry. I formed it for my friends and family. It has raised $10 million. And by coming in, you'd be pooled with all the prior investments. And so you'd be getting equity at an average valuation of 50 million. So there is an opportunity if you want to come in through the SPV to get equity at a $50 million valuation right now. Or if you want to be at the top of the liquidation stack, you could go into the note and get the discount to whatever equity financing we do next.
Got it. I think the minimum ticket size for the SPV, Paul?
Well, with the SPV, we can do smaller tickets. My preference is for people to come in with $50,000 or more. So you have to be an accredited investor, first of all. The reason for the $50,000 is I like to know every investor, and I like to spend an hour with them. And if I spend an hour for small checks, I wouldn't have enough time to do my job. So, but as I said, I can make exceptions. So, if you're committed to investing and you can, and you don't need a lot of explanation, then just reach out to me.
Got it. Might have time for one final question. Anybody have any additional questions for Paul or Jeff? Feel free to type it in there or raise your hand and we can call on you. Otherwise, I would love to thank Paul for his presentation and overview of Angiex. We appreciate your responsiveness to the poll that we had earlier for the investors. Again, if anybody's interested in meeting up with the Angiex team, thank you, Emily. You can DM Paul directly or Jeff Sacker, who's on the call as well. But with that, I'd like to thank everybody for joining us. For those of you who have expressed an interest, we will be following up with you later today or tomorrow. We will send out the deck as well as a link to the recording for those of you who may have showed up later if you want to share it with a colleague or a co-investor. But thank you guys very much for attending and have a great day.